probably seen it around. Our next talk will be on September 24th. It'll be Edgar Perez Lopez, and he will talk, his title is called May the Fields Be With You, Microwave Power and the Plasma Frontier. So make sure that you keep an eye out for the posters, check out what talks interest you. And then we also have our other program, Modesto Area Partners in Science, MAPS. So we have Wild Planet Day coming up. We have some really fun activities planned for that event, so make sure that you attend. And then of course we have our Great Valley Museum. We have our hours listed on here, and then Science Night every first Friday of each month. So I just wanted to kind of run through that before we get started. And then let's just go ahead, let's pull up our presentation. And then I'll hand it over to Professor Danielle Hicks. So let's do slideshow. Where am I at? Oh, right here. Yep. You clicked. I'm like, what did I do? Oh, there we go. And then take this out. We'll set you up with your clicker. Perfect. And then turn this on. Good? Okay, yep, perfect. On the laser. All right, let's give it up for Danielle Hicks.
Hello, so welcome to Science Colloquium. I'm Danielle Hicks. I'm super excited to be starting us off this semester with our colloquiums. And before I get started on my talk about stress in cells, I just wanted to give a little preface to research, okay? Because I'm going to be presenting the data that I collected when I was a master's student at University of the Pacific. And you know, it might sound fancy and really complicated to do research and everything, but I just want to share that I had a lot of imposter syndrome before I went and got my master's, and I had a lot of anxiety about the whole research aspect and the idea of completing a thesis. And so I just want to share just really briefly kind of about that. And I was really excited going into my master's to, you know, teach, and I was really excited about taking classes and learning more. But then like the idea of the research element was very overwhelming. I knew it was going to be a lot of work, and I didn't know if I'd be able to do it. But as I hope you are going to be able to see through my presentation, I actually fell in love with what I ended up doing, and I loved the research and just thought it was so fascinating. And obviously I ended up coming into teaching, but I still enjoy the research so much. And so I just encourage you, you know, as you're in school, it can be stressful and you might not feel good enough. You might feel like it's too much for you, but I encourage you to just keep pursuing things that feel hard, things that might give you anxiety. Keep going after it, and you never know what's going to happen, right? It gives you so many opportunities, and you might find something that you love that you never thought you could, okay? So with that, I'm going to be talking about zebrafish and cell stress. And so the title of my presentation is Stressed Out Cells, What Zebrafish Are Teaching Us About Cell Survival. So as just a kind of overview to this talk, I'm going to be answering the question, what is cell stress, first of all? And then we're going to talk about how cells respond to certain types of cell stress. And then I'll be presenting the research that I conducted as a master's in biology student at University of the Pacific. So I'll share some of my techniques and then the results of that data. And then I'll give a discussion of our conclusions from that project and then the future directions that are still happening for that project, okay? Okay, so to first address the question of what is cell stress, okay? We're all human beings here, right? And we probably have a pretty good understanding of what stress is as just kind of people, right? You're all in school probably, or you have jobs, right? Just being a person is stressful, okay? Having deadlines, having any responsibilities in your life, that can cause anxiety. And you guys are probably all really well aware of what that feels like. Things that you do to cope with stress that are maybe healthy and maybe things that are not so great in your life, right? But what I want you to think about for understanding this talk and this research is the fact that cells experience stress, and we have lots of stress that actually happens at all of these different levels, okay? So you're seeing kind of the different levels of organization here of a human being, and our organ systems can experience stress. Organs experience stress, tissues, cells, even parts of our cells that we call organelles, okay, they can even experience stress as well. And so really what I'm going to be focused on is stress at the organelle and cellular level, okay? So, and we can have all kinds of different causes that result in cell stress or organelle stress, okay? Some examples might be things like temperature, right? If your cells get too hot, that's really stressful for them, right? If they don't have enough oxygen, that's really stressful. If they don't have enough nutrients, that's a stressor. So there are lots of different things that can cause cell stress. But we're going to be talking about one specific example of that in a second. Okay, so essentially cell stress, we would say, is a disruption of homeostasis in the cell. Okay, so something has gotten off balance in our cell, and that's a problem, right? We don't want to be off balance. Okay, and so if you are maybe in biology, or certainly if you're in a physiology class right now, I know that you have seen that picture so far this semester. And this is kind of just the textbook picture that shows what happens when something is off balance. And so it kind of uses the teeter-totter there, right? We want things to be in balance, and if something tips us to have too much or too little of something, that's an imbalance, and that's kind of a problem, right? And so your body is going to have a way of dealing with that imbalance to try and restore balance, right? Does that make sense? And so I'm not going to go through all these different components, but the idea here is that when there's an imbalance, okay, in homeostasis, you have mechanisms or pathways that get initiated in order to restore that balance. Okay, so a typical example of this would be things like body temperature, right? We're kind of winding down on the summer months here, right? And if you've been outside for too long one day and it's 100 degrees, your body temperature is going to start to rise, right? It's uncomfortable. That's actually stressing out your cells. And your body has a way of detecting that and responding to that, right? So you have thermoreceptors in your body. They're going to be alerted like, oh, it's too hot, right? Our body is too hot right now. They're going to tell your brain about that. And then your brain is going to do what? Cause you to sweat, right? And so that's kind of the response. Sweating then, your brain's gonna tell your sweat glands to really sweat, and that's gonna help to cool you down. And in doing that, we've kind of completed the cycle here, and we've restored the balance, right? Body temperature was too much, and then we cool it down so that we restore homeostasis in the body, okay? And so that's kind of looking at homeostasis and a pathway to correct that at a big level in the body with multiple organs being involved there, right? But this also happens within our cells, okay? So when a particular cell experiences stress, it's gonna have ways of noticing that stress, and then there's gonna be pathways that get activated in order to deal with that stress and kind of return us to our state of homeostasis and balance, okay? So the particular type of cell stress then that you need to kind of understand for my research was protein homeostasis, okay? And what we were looking at was what happens in a cell when there is a loss of protein homeostasis. And it turns out that that is actually incredibly stressful for our cells to deal with, okay? So I'm gonna just recap a little bit about proteins in case it's been a while, but proteins are incredibly important to our cells and to our body, as I'm sure you're aware, right? We have different biomolecules that make up our cells and our tissues, right? We have carbohydrates, we have our lipids, we have nucleic acids, and then we have proteins. And they're all really important, but proteins in particular, we often talk about in biology as kind of performing the majority of cellular and body functions, okay? So proteins are incredibly important. And this picture here is meant to show you some of those major functions that proteins play for our bodies and for the cell. So the one here we're seeing is in digestion, right? So anytime your body needs to perform a chemical or metabolic reaction, an enzyme is helping to facilitate that, okay? And enzymes are proteins. And so reactions that are chemical reactions in our bodies are done by proteins. And that happens all the time in all of your cells, okay? I'm sure you are aware of the fact that muscles rely heavily on proteins to function, right? So your muscle cells are predominantly just a bunch of proteins, and that allows us to move things in our body, to have big movements, and to contract muscle, right? And our immune system relies on proteins, right? Antibodies are a class of proteins, so to keep you healthy, to keep you free from viruses and pathogens, we use proteins for that. Structural support, a lot of proteins provide structure to our tissues, right? Things like collagen. provides structure to our cells and tissues, and then many of our hormones are proteins. To even express our DNA, we use a lot of different proteins for that. To move things around in our blood, to transport oxygen and other things in our blood, we use proteins, and on and on and on I could go, right? This is not a comprehensive list. This is just a few of the many, many, many things that proteins do, right? And so when we think of proteins, all proteins are going to have a particular, very specific shape or conformation, okay, that allows them to do their job, okay? So a protein, right, is a strand of amino acids, and we fold a protein up into a very particular shape or conformation so that it can do its job. And if it loses that shape, it is no longer functional, okay? So it's very highly important for a cell to keep a protein's shape so that it can do its role for that cell or for the body, okay? So I'm gonna bring us now to one of the major organelles that processes and folds up our proteins, okay? And that is the rough endoplasmic reticulum. So you're seeing the rough endoplasmic reticulum here. It lies right outside of the nucleus, and it is the site of protein synthesis, folding, and processing for any protein in the cell that is either going to be secreted from that cell or any protein that is going to become a part of the plasma membrane. Okay, so those are two of the major destinations for proteins that are being processed and folded in the rough endoplasmic reticulum, right? So we'll take a look at that here, that pathway real briefly. So you're seeing here mRNA. It's been copied in transcription from DNA in the nucleus, and that mRNA leaves the nucleus, and it's going to find a ribosome, which is the organelle in the cell that makes proteins, okay, and does translation. And if that protein is going to end up being released from the cell, right, or become a part of the plasma membrane, that's going to kind of shuttle that ribosome and the mRNA to the rough endoplasmic reticulum. The ribosome attaches itself there, and then the rest of that protein synthesis is going to be completed inside the rough ER, so inside the lumen of the rough ER. All right, and you have helper proteins inside the rough ER that we call chaperone proteins that are going to be there to help fold all those proteins into their proper shape. Okay, so we have lots of helpers that help get those proteins into their perfect conformation so that they can go do their job in the body. All right, and then on we go. That those proteins will go to the Golgi apparatus, and then eventually they'll be either released from the cell, like you're seeing here, or you're going to embed those proteins in the plasma membrane. Okay, so that's kind of just an overview of the pathway of proteins that are being processed in the rough endoplasmic reticulum. Okay, now that accounts for approximately 1/3 of all protein synthesis that is occurring in eukaryotic cells. We're eukaryotes, so that's in our cells as well. 1/3 of protein synthesis is happening in this kind of pathway here. Okay, so that means that 1/3 of proteins are going through the rough ER. And because of that, you can imagine that the rough ER plays a major role in regulating protein homeostasis. So it's a major player in making sure that all these proteins are processed correctly and can move on and go to the right part of the body to perform their job, okay? Now, we can experience stress here in the rough ER, okay? And so we're going to talk about that. It's called ER stress, all right? So there are, well, first of all, the ER is kind of a sensitive little organelle, okay? It's doing some really important functions, and so it has to have just the right conditions to be able to do all that it has to do. Okay, it's kind of a sensitive organelle, and so there are, many numbers of stressors that can actually impact functioning of the rough ER, right? So it's sensitive to low energy levels, low nutrient levels, low oxygen, okay, and increased protein demands. There's lots of different things that can become a stressor on this organelle. And what happens with stress is that sometimes the protein folding demand exceeds the ER's protein folding capacity, okay? And so when that happens, we call that ER stress, right? So there's too many proteins that we're trying to process and fold, more than we can handle in the rough ER, and that's a source of stress, okay? And we're gonna see what happens then in response to that is that there gets to be kind of a backup here in the rough ER, okay? We get the accumulation or aggregation of proteins here, okay? Those proteins can either be misfolded, meaning that they have been folded up into the wrong thing, okay? Those can be toxic to our cells, or sometimes it's just that the proteins are just unfolded. They haven't been processed yet, and so they just kind of remain in this unfolded state. Either way, that's bad, okay? We don't want this backup of proteins because that can be, again, toxic to our cells, and it can lead to diseases, and it can even have toxic effects on other neighboring cells. And so we don't want that, okay? That protein aggregation, you might have this situation going on at your house right now, maybe, right? And we can think of stress that is caused by, piles of laundry at home, okay? Right? Maybe, you've run the washer 3 times today, you also ran it 3 times yesterday and the day before, and you have loads and loads and loads of clean clothes, but what happened? They're unfolded, right? And they're gonna keep piling up, okay? And, similar to how that maybe stresses you out at your house when that happens, right? When you have protein aggregation in the ER, that stresses out your cell, okay? It is not a good situation. So, as you might imagine, ER stress is related to a lot of different diseases, okay? We see this as actually a hallmark of many neurodegenerative diseases, okay? And so I'm showing you some of the most famous ones that affect a lot of people all across the globe here, things like Alzheimer's, Parkinson's, Huntington's, and ALS, but there are lots more as well. We see that an accumulation of misfolded proteins in the ER and sometimes elsewhere in the cell or even outside of cells is happening in many of these neurodegenerative diseases and leads to the progression and the severity of these diseases, okay? But we also see ER stress and accumulation of misfolded proteins in other types of diseases as well. So we see in lots of metabolic diseases, things like type 2 diabetes, the non-alcoholic fatty liver disease, those are a couple big examples, but also cancer, okay? So we see this happening in lots of diseases where you get this backup of proteins inside the cell, and that being a big stressor for cells in disease, okay? And what can happen is that the response to this protein aggregation is that can lead to death of the affected cell, okay? And in this case, it would be a programmed cell death, okay? We call that apoptosis, all right? So it's actually kind of a decision that a cell would make to preserve the integrity of the tissue or the organism, okay? So sometimes when you have so much stress in a cell that's kind of irreversible, it's so severe or so chronic, it's actually better to tell that cell to just die, all right? And that's actually gonna be helpful overall to the tissue or to the organism, okay? And that sounds kind of bad, but it's a protective mechanism and it can help prevent cancer too. But when we're specifically looking at our neurodegenerative diseases, when you have cell death occur in a neuron, that's especially problematic, right? And because we don't regenerate typically our neurons, right? We don't have mitosis happening in our neurons, and so when you lose a neuron, you're losing some neural function for the body, right? And you can make it years, with some neuron death, that's okay for a while. We have backup pathways, but eventually you're going to see an impairment in cognitive function, right? Maybe memory loss and or even motor functions as well, where you can lose that communication or affect communication to your muscles, okay? And all of that can lead to death ultimately. So when we're thinking of ER stress and disease, we want to look at, how can we maybe manipulate the stress, reduce the stress ultimately, and prevent apoptosis of our neurons especially, okay? And so that's what my research especially focused on, was the relevance to these neurodegenerative diseases. And it turns out that there are actually some small molecule drugs that have already been discovered, some are already approved for use and for prescription, that treat some classes of neurodegenerative diseases, okay? And they're working, we think, by reducing ER stress and kind of improving, the survival of neurons and helping to kind of reduce the progression of these different neurodegenerative diseases, okay? The problem, though, is that a big problem in pharmacology is just that sometimes we don't really know how those drugs are working. So you can discover that a drug seems to work. It doesn't seem to have many negative side effects on the patient, right? And it seems to improve disease outcome. And so it can, get approved for use, but that doesn't necessarily mean that we know all the ways that it's really functioning inside the cell at a molecular level, okay? And so we need a better understanding of these particular drugs, right? And we need a better understanding of ER stress-related pathways, because there are potentially a lot of drugs that we could be using to reduce stress in these different diseases, or even develop new drugs to target certain parts of these pathways, okay? So now we're going to take a look at how cells are responding to ER stress, okay? So the quick answer, the nice little simple answer, is the unfolded protein response, okay? The UPR. I'm going to be calling it that, but it's this response in your cells that gets activated when you have a bunch of unfolded or misfolded proteins, okay? And the slightly longer answer is this here, okay? So I'm not going to be going through all of that today, okay? You're welcome. But if you know me, you know I love a good cell signaling pathway. And this is actually what drew me into this research, is when my advisor got up and started drawing the pathway on the whiteboard, and I was like, what is happening? I've never seen something so complicated, but I loved it. I thought it was so interesting, and I had no idea that your cells could actually have such intricate and complex pathways, and so that's kind of what I initially fell in love with. But this is even a simplified version of the unfolded protein response. But if you kind of look here, you're seeing A, B, and C. There are 3 kind of branches to this pathway, and I'll talk about those in a little bit. But the research that I did was really mostly pertinent to this one branch here that we call the PERK pathway of the UPR. I'll be going into detail on that in the next couple slides. But for now, I want you to think about the unfolded protein response, or the UPR, as being kind of analogous to pulling the fire alarm when you notice that there's maybe a problem in a building that could potentially lead to a bigger fire, right? You see something is sparking, and you want to tell everybody that, hey, we should probably get out of the building, right? We should probably deal with this issue before it gets any worse, because it has the potential to blow everything up, right? So you'd pull the fire alarm and kind of pause everything that's happening so that you can deal with the issue, right? So that's kind of what the unfolded protein response is like at this general level. And if we go back to our laundry analogy, okay, in the case of you have piles and piles of clean but unfolded laundry, something that you might do, which is kind of like the unfolded protein response, is shut down your washing machine. Tell your whole family, we're not running the washing machine for the rest of the day, and we're all gonna get on board, and we're gonna fold the laundry, and we're gonna put it away, okay? We're gonna deal with the problem that we have and not make it any worse, right? That's basically what the unfolded protein response is doing, but inside your cells, okay? So now we're gonna look at that unfolded protein response, and I'm bringing you back to these 3 protein pathways here, okay? So we have 3 different pathways, and what you really need to know is that the overall outcome of the signaling here are these 3 main things. First things first, we're gonna decrease protein synthesis, right? If we have ER stress, let's stop making new proteins for the most part, right? That kind of makes sense. We don't want to contribute to making the problem worse before we've dealt with the problem. The next thing is that we're going to actually increase protein degradation, okay? So that's especially helpful for all those misfolded proteins, right? If you have these proteins that are in the wrong form, we want to degrade them and get rid of them because of their potentially toxic effects, okay? And then the last thing here is we want to actually increase protein folding. So we're going to make more of those helper chaperone proteins that I mentioned before, because they help to fold unfolded proteins. And if we make more of them, then maybe we can actually salvage some of the proteins that we have in there that are that are unfolded, right? So if we can use them, then let's do that, right? So these are kind of the 3 main outcomes of the signaling of the unfolded protein response, okay? The goal here is survival, right? The goal is to actually return the cell back to homeostasis, particularly return the rough ER back to a state of balance, back to homeostasis, and allow that cell to survive, right? That's the goal, okay? And that's what normally happens, right? Maybe your cell is kind of stressed because you haven't eaten in like the whole day, right, and it's 5 p.m. and you're starving, that's going to stress out your cells, and that could potentially lead to some ER stress in some of your cells. And you would activate the UPR, and in a case like that, you know, you're not experiencing like a disease state, that's going to promote survival. And so that's what happens most of the time. However, the problem can be when there is either severe and or chronic ER stress, okay? And so when that happens, and we're thinking especially in diseases here, the cell might determine that the problem is kind of irreversible, right? There's actually so much ER stress, too many proteins to really be able to deal with. It's not gonna be possible. And so that's gonna send the cell in towards that death apoptotic program where it's going to kill off the cell, ultimately to protect the overall tissues and nearby cells, okay? So there's pretty much always this decision point here, okay? Can we promote survival? If so, let's do that. But if not, right, we might have to lead to cell death, okay, for the good of the organism, all right? So that's kind of always a decision that the cell is going to be making. Okay, and so like I said, my research focused on PERK, and so I am going to spend a little bit of time going through this pathway. If you can hang in there with me, it'll make my actual data make a lot more sense. And I love these pathways, so I hope you can follow, right? So I'm looking at this one pathway of the unfolded protein response. And ER stress is going to activate a protein in the cell called PERK, okay? PERK is a kinase type of protein. So that means that it is going to change other proteins in the cell by adding a phosphate group onto other proteins. And that's what kinases do. And so the target of PERK activity is this protein here, okay? It's proteins acting on proteins, acting on proteins. PERK puts a phosphate group on this protein here called eIF2 alpha. It's an initiation factor, okay? So it's eukaryotic initiation factor 2, the alpha subunit, and essentially this is a protein that is necessary for protein translation in the cell. So to make mRNA into protein, you need eIF2 alpha for most of that to happen, okay? And so when you phosphorylate it, it actually kind of deactivates it. And that's going to stop or attenuate most protein synthesis in the cell, okay? And so that goes back to that one major function, right? Like, let's shut down protein synthesis for the time being so that we can deal with the problem. And that's what's happening here, okay? With phosphorylating eIF2 alpha, you're shutting down protein synthesis for a period of time, okay? When eIF2 alpha is phosphorylated, though, there are some proteins that will actually be activated and come about, okay? Otherwise, they wouldn't be. But one of those is ATF4. So ATF4 in an unstressed cell is not very present, but when you get stress, it comes alive, okay? And it's going to start being more active. It's a transcription factor, so that means it can go into your nucleus and it's gonna cause the activation of genes that are gonna help your cell survive, okay? So we're gonna promote chaperone production, right? We're gonna make more of those helper proteins. We're gonna promote things that are gonna degrade the misfolded proteins. And really what you need to know is that ATF4 is involved in promoting things that will help that cell survive. And we think of both of these as being involved in kind of the early stage of the UPR, okay? So the first 4 or so hours are especially kind of dedicated to, you know, trying to help that cell survive, okay? Now, what can happen is you eventually want this pathway to turn off, right? You don't want it on forever, okay? That would not be good. And so as time goes on, you're gonna activate a protein called CHOP, okay? As you might infer, CHOP is not super great for your cells in terms of survival, okay? And as you see here, CHOP is involved in promoting apoptosis, so it has pro-apoptotic targets, okay? So it's gonna be involved in kind of leading the cell towards cell death, okay? And that's because if you've been in this pathway long enough for CHOP to be activated, it might indicate that the cell is kind of a goner, right? It might indicate that things are irreversible and we need to start thinking about cell death rather than survival, okay? So that's kind of the PERK pathway. Now, the actual proteins that I studied were inhibitors of this pathway in that they turn this pathway off. Right? So you turn this pathway on, you try and promote survival, but at some point you got to turn the pathway off, right? And here come in the two proteins of my research, which are called GADD34 and CReP. They have super fun names, okay? So GADD34 and CReP. And these two proteins are activated by both ATF4 and CHOP, so both of these guys can activate GADD34 and CReP. And GADD34 and CReP are phosphatases, so they are eIF2 alpha phosphatases. What that means is that they are doing the opposite of a kinase. So kinases and phosphatases have opposite functions. So kinases, remember, add a phosphate group onto a protein. That means that a phosphatase is taking it off. Okay, and so GADD34 and CReP can each bind to this protein here called protein phosphatase 1, PP1. And together they can help take us this way to the right, with this arrow down here. So they're going to help remove that phosphate group from eIF2 alpha. And what is that going to do? It's going to restore protein synthesis in the cell, right? Because this pathway here was kind of about slowing down, stopping the protein synthesis in the cell for a period of time. But when we go back to normal functions, we've got to resume protein synthesis. So it's kind of bringing us back to the normal rate of protein synthesis in the cell we're done kind of with the UPR. It turns off the UPR essentially here. So these are eIF2 alpha phosphatases. Again, they're dephosphorylating eIF2 alpha and restoring our protein synthesis, okay? So these two proteins that were the focus of my research, these eIF2 alpha phosphatases, there's a lot that we don't really know about them, okay? So there's a lot of research that has been done on them, but it's not really clear exactly what they do at all times. So we know from other labs and also other students that were colleagues of mine in my lab that GADD34 is strongly induced by ER stress. So we know that it kind of acts as what we think is the primary negative feedback inhibitor of the PERK pathway. Okay, so we know in stress cells it is absolutely active, but we've also found that CReP is involved in stress as well. Okay, so maybe it's playing a bigger role in stress than we once thought. Okay. And we know that CReP seems to have maybe more of an important role in kind of a non-stressed cell. But we also know that GAD is involved in non-stressed cells as well. So as I'm kind of showing here, there's a lot of overlap and potential redundancy, but we know that there are differences as well, okay? We know that CReP is also necessary for survival. So studies have been done in mice. If you have a mouse that doesn't have GAD, you wouldn't even know, okay? It's kind of undetectable. If a mouse doesn't have CReP, it will be born, but it doesn't live too long. It has problems with its red blood cells. It has growth retardation, so it doesn't really grow the way it's supposed to. And that tells us that CReP is, more necessary for survival than GAD probably is, okay? And so the point of my research was kind of, first of all, to determine what are the more specific roles, what's the regulation, what are some of the similarities and differences of these two proteins in the PERK pathway of the UPR, okay? And we used zebrafish as the model organism for this research, okay? So zebrafish are maybe a model organism that you aren't super familiar with. You're probably familiar with, fruit flies maybe, or mice being model organisms, but these tropical fish are actually really great model organisms, and they're excellent for the pathways that we were trying to study. The first reason is because they actually have both GADD34 and CReP homologs, okay? So that's perfect because then we can study their differences, their similarities, and their interactions, okay? Other organisms like fruit flies and yeast and nematode models, they either don't have either of these two proteins or they only have one. And so obviously that's not super helpful, right? The next thing that makes these great model organisms is that they produce large numbers of embryos that are actually external to their bodies. So they will spawn, and the eggs and sperm are laid externally, and so fertilization happens externally in the water, and you can just collect all those embryos and perform studies on them, okay? So that allows you to conduct studies that have large numbers of organisms, okay? You can get high sample sizes relatively easily because every time you spawn them, you can potentially get hundreds and hundreds of embryos produced that you can run tests on. The next neat thing is that they are transparent, okay, as you're seeing here. So in that early development of the embryo, you can see right through it. So that allows for really easy imaging assays, and that's what we took advantage of for my studies, okay? And they develop really fast. So here you're seeing the zygote, so that's the actual cell that will become the zebrafish right there. And that's about the first 30 minutes or so, it looks like that. And then it will start undergoing mitosis and division. And at just 24 hours later, this is what you see, okay? Pretty impressive development, right? In one day, you've got a brain, you've got eyeballs, you have a full like digestive system, and you have a caudal fin, which is probably the least impressive, but that was actually the important part to my research. I conducted my tests on the caudal fin here, okay? And so you can conduct these really detailed in vivo studies here, okay? So the caudal fin was important to my research because these cells here, in making this fin for the fish that will end up looking like that, these cells here are actually pumping out a bunch of cartilage, okay, and a bunch of extracellular matrix material. All of that is protein, a lot of protein that has to get exported from the cell. So you have a ton of proteins going through that rough ER pathway for secretion, okay? So essentially in this early development, these cells here in the epidermis are highly stressed, okay? They're like already kind of on the verge of being too stressed out, and you can give them a drug that makes the stress a little bit worse and tip them over the edge and make them a little bit more susceptible to death. And so you can see there how that's an excellent model for some of these neurodegenerative diseases in humans, right? It's very, very similar, right? You have high levels of stress, the UPR is kind of already on, and if you make them a little bit more stressed, they might die, and then we can see what rescues them, okay? And so that's how we did my studies. And also these tails lay flat. So they're pretty flat, and you can see kind of everything there and look at the cells there. Okay, so somebody in 2011, Payati in his lab, found this really interesting thing about that caudal fin in the zebrafish, okay? So they took a look at what happens to these cells when you stress them out, okay? So we give them some stressor, and they noticed that there were cells dying at this early time point following that stress, okay? So at about 4 hours after that stress, they noticed that there was this acute cell death. So cells would die, and the way that they would be targeted for apoptosis was through these genes or proteins here. So P63 protein, and then also PUMA, okay? And I'm not gonna have time to get into the details there, but basically they are proteins that can trigger a cell to undergo cell death, okay? And so that was what was happening here at that 4 hour time point. But then if you waited longer, there were other cells that would be dying at 24 hours after stress, okay? And that they called kind of the chronic cell death, and that death was mediated by CHOP. Do you remember CHOP? Okay, so CHOP was part of our PERK pathway, right? And so that was that protein that if you kind of have the UPR going for too long, right, CHOP's gonna start to accumulate, and it's gonna start to tell your cell to die because the ER stress is too chronic, right? It's been around for too long. So that's really interesting, right? That there are two different ways of telling the cell to die, and it's all just based on how long it's been since the stress, okay? So my specific project was really to look at the roles of GADD and CReP in these two different types of cell death in the caudal fin of the zebrafish embryo. Again, as a model for looking at neurodegenerative disease, okay? And so the question was, what are the roles of GADD and CReP in these two distinct genetic programs of cell death? And so we did loss of function studies and gain of function studies, okay? And so I'm gonna talk about that real briefly. So loss of function means that you're knocking out the function of a protein, right? So there, the idea was like, okay, if we knock out GADD and CReP, or both of them, and we kind of inhibit this ability to dephosphorylate EIF2 alpha, what's gonna happen, okay? And our hypothesis was really that if you're preventing dephosphorylation, then we're gonna have increased levels of the phosphorylated EIF2 alpha, and essentially kind of have the UPR in this early pro-survival phase for longer. So potentially, we think that if we lose their functioning, then we can promote survival by keeping the eIF2 alpha phosphorylated for longer, okay, and kind of stall the progression of the UPR, prevent it from progressing down into CHOP, okay? That's kind of the idea. And so we did loss of function studies by something we know as morpholinos, and then also with a drug called salubrinol. So real briefly, morpholinos are a way that you can prevent a functional protein from being made, okay? So normally what happens in the cell is you have your pre-mRNA, introns get taken out, and that leads to the exons joining together, and you'd get a functional protein from that, okay? But we can add in something that actually blocks that from happening. So now you can no longer chop out this intron, and now it stays around. The spliceosome can't take it out, and that results in a non-functional protein. Okay, so this is a way that you can inject the morpholino into the zebrafish and knock out that protein eventually, okay? And so the experimental design here was that you take, or I would take these embryos when they look like this at the 1 cell stage, and I would inject them with the morpholino, okay? Very tedious process under the microscope. But then as the zebrafish develops, it has that protein knocked out in all of its cells, okay, as it develops. And so then we'd let them grow for about 24 hours, and then we would stress them out with a drug called thapsigargan, okay? So that hits them with some stress, and then 4 hours at that early time point, and then at that 24-hour time point, we would take a look at then what's happening in the tails, right? Do we have more or less apoptosis? And so to kind of look at that, we did an apoptosis assay known as acridine orange staining. Basically, it fluoresces in cells that are undergoing cell death. So the more bright green, it's really pretty, but it means that a cell is dying there, okay? And then we would image that under the confocal microscope and take a picture. And then I got to do this really fun thing where I would count manually the number of apoptotic cells in all of the tails, and that was many hours and days in the dark, counting and recounting. And eventually we kind of averaged out the number of apoptotic cells in each of those groups. And so on the next few graphs, that's what you're going to be seeing. Before we get to those, just to show you here that thapsigargan does indeed induce apoptosis, okay? So you can see that at that 4-hour time point and the 24-hour time point, you have lots of bright cells here, and so that does induce ER stress, and so we can see what rescues that from happening. So the first knockout that I did, the first loss of function, was with GADD34 with the morpholino. And the results we got here are that at that early time point, there was no significant difference, okay? But at 24 hours, we actually saw a decent reduction in the number of cells that were dying, okay? And so that kind of supported what we were thinking, that supported the hypothesis there. That stalled the progression of the UPR and allowed for more time to promote survival. With the morpholino targeting CReP, we actually didn't see a significant reduction in cells that were dying, okay? So CReP alone did not seem to be able to reduce the cell death at either time point. However, when we did a double knockdown, so when we knocked out both GAD and CReP, we saw a statistically significant reduction in protein synthesis at both the 4 and the 24-hour time points, okay? So that showed that if they're working, if we kind of work together and target both of them, we are going to be able to promote survival at both of these different time points for cell death, okay? All right, and then we kind of did that same experiment with the double knockdown of GAD and CReP with this drug called salubrinol. So this is a drug that blocks both GADD34 and CReP at the same time, okay? And we had a pretty similar treatment here. I didn't have to inject anything here, but we did incubate them for 2 hours in the salubrinol and then did the same thing. Hit them with stress and then waited for 4 hours and then 24 hours and assayed them for apoptosis. And we saw similar results to the morpholino studies here, okay? So with the salubrinol treatment, where you're knocking out both GAD and CReP, we saw that at the 4-hour time point and the 24-hour time point, there was a statistically significant reduction in the stress, okay? And a reduction in cells that were dying. So that confirmed those morpholino studies there, okay? And then we did a little bit of gain of function studies. It turns out this was kind of harder than expected. And we really did not know what was going to happen here. So what happens if we make more GAD or more CReP or both? You know, we kind of predict that maybe we'll have the opposite effects, right? Maybe this will actually make the cells die more. But we really didn't know. Okay, there's a big kind of question mark here. And so with the gain of function experiment, I was able to complete this with GAD. And so I injected mRNA for GAD into the embryo and then went through the same series, stressed out the cells and then assayed them for apoptosis at the 4-hour and 24-hour time point. And looking here, we saw some really interesting results. So GADD34 gain of function, so when it has lots of GAD, it actually promoted survival at this early cell death time point, okay? And that was totally unexpected. You know, I think we definitely thought that it was gonna make things worse, but it turns out this actually had a protective effect here. And we don't really necessarily know why. We have some ideas, but there definitely needs to be more research done to kind of figure out why that is the case, okay? Now, there wasn't really any statistically significant difference there at the 24-hour time point. And I wasn't able to do that study with CReP because CReP is quite difficult to work with. There's just always problems that happen with CReP in research, it seems like. All right, so for the discussion, to kind of come back to the question, right, we were looking at what are the roles of these two proteins in each of these programs for cell death, right, at the 4-hour and the 24-hour time point, okay? And we can come to just the summary table. So this summarizes the results that I got. We saw, you know, mostly I did the loss of function studies, and we saw that knocking out GAD promoted survival, okay? That's what the star is showing at that chronic time point of cell death mediated by CHOP. CReP knockout really didn't have any effect either way, but then when you knocked them both out, either through the morpholino or with the drug Salubrenal, we see a pro-survival effect at both time points. So that tells us that this is a really strong candidate, right? The knocking out of these two proteins might promote cell survival in neurodegenerative diseases. And then this kind of question mark here of GAD overexpression seems to also be protective, but at that acute cell death, okay? So the conclusions we kind of make are that GADD34 is still, we still have a lot of unknowns, okay? It probably is interacting differently with these two different programs for cell death. And loss of CReP alone seems to be insufficient to alter the dynamics of the UPR. But when we lose all of that eIF2 alpha phosphatase function together, that seems to protect cells from both acute and chronic ER stress-induced apoptosis, okay? And the conclusions then are that manipulating these proteins is, you know, definitely beneficial to minimizing ER stress-induced apoptosis, as I said, okay? So it seems that when you have a cell that is under chronic ER stress, okay, longer term issues with ER stress, we want to inhibit the eIF2 alpha phosphatases, okay? And kind of let that pathway stall and kind of stay in the early stage for longer. And potentially in other cases, maybe in more metabolic diseases or cancers, maybe we should upregulate GADD34. Maybe that promotes self-eating, which we call autophagy. And maybe that's the way that it's protecting these cells against that early time point of death, okay? But again, we don't know all of those conclusions for sure, but we did get a little more information there. The lab that I worked for is, of course, still active and still doing lots of studies. We need to do the CReP gain of function that I wasn't able to do. And then they've actually developed, since I've been gone, knockout zebrafish lines. So genetically altered zebrafish that do not have the gene for GAD or CReP, and then they also have the double knockouts. And so those are excellent for conducting studies because they already have those genes with a loss of function, okay? And so you can do a lot of stuff there. And then there's also potential to study these pathways in other types of diseases. So looking at metabolic diseases, like the non-alcoholic fatty liver disease. And you can do that in the zebrafish embryos as well. All right, and then I haven't done the wet lab research there. I did work a little bit after my master's thesis and continued some of those projects. And then I was also kind of lucky enough to be able to contribute to writing and researching for this very first review paper that was published on these two proteins. There had not been a review paper yet. And so if you're interested in this stuff, if you like these pathways, I put the QR code there. You can find it. It's an open resource journal. And if you like the research, I'm more than happy to answer any questions you might have about getting your master's. All right, I do want to acknowledge my research advisor, Dr. Douglas Weiser, and my other advisors at UOP, Dr. Rishnik and Kudyakov, my colleagues, and then all the funding that made all of this possible. It made it possible for me to end up here as an instructor. I'm very appreciative to University of the Pacific and the fellowship I got there to do research, as well as the federal funding from the NIH. Quite important to be federally supporting this type of research. There's a lot that goes into understanding disease and treatments for disease, and it does not happen quickly. It takes years and years and thousands of people to be able to come up with therapeutics and understand why they work. And so I encourage you to do what you can to continue to support federal funding of research. And with that, I will answer any questions that you might have. All right. Yes.
Do you know so it kills off the cells because they're damaged or falling apart or something. Do you know since you stalled them out, do the cells ever fully recover, or are they permanently damaged and cause cancer?
Yeah, that's a great question. We don't quite know, but it does. It's not like you can, if there's some underlying cause of the stress in a neuron, for example, it's not that these drugs would necessarily be able to cure that at a root level, but it can keep the neuron alive for longer, right? For maybe more years. It might have compromised function, but at least it's functioning a little bit. But it doesn't mean it'll survive forever though, but it can, essentially these drugs would be looking at prolonging the progression of Huntington's onset, or the cognitive effects you'd get with Alzheimer's, or the progression that you'd get muscularly with ALS and Parkinson's and stuff like that. So it's not a cure, but it is kind of a treatment that would prolong severity and symptom onset. Yeah, good question. Yes, in the very back there.
Cell death and protein aggregation related to the calcifications seen in neurodegenerative diseases such as Parkinson's?
Yes, so I might not quite know what the calcification you're referring to exactly, but calcium does, I didn't really talk about it at all, but calcium absolutely has a really important role here. So the ER, especially in the smooth ER, is where your cells store calcium, and for protein folding to be done properly, you have to have the right amount of calcium in the ER for that all to take place. So when you disrupt calcium homeostasis, that is a big trigger for ER stress, and then for those pathways to get turned on and to promote death. So actually that drug, that thapsigargan, the one that we stress the cells out with, that's actually how it causes the stress, is by inhibiting the calcium pumps on the ER, which disrupts the calcium balance in the cell, and that's how it causes that pathway to get started and lead to death. Yeah.
Do you believe genetics plays any role in how slow or how fast apoptosis occurs?
Yeah, absolutely. Yeah, so there could definitely be, if you at a genetic level have any mutations or any way that some of these genes in any of these pathways are not able to be made properly, right, to be functional, that would lead to impairment of any of these pathways and therefore the response to stress. So yeah, absolutely. There are certain genetic, some people, for example, there are people who are born with a defective CReP gene, and we see that sometimes. It's pretty rare, but it can happen. So yeah, absolutely. Any other questions?
We don't have any other questions. I'm sure that you'll be happy to stick around a little while.
Yeah,
People
absolutely.
Come up to you. But yeah, thank you guys for attending our first science colloquium. We'll see you again on the 24th. Go to your title slide.
Oh, yeah.
Because I'll send it to Nino's for the newsletter.
Perfect.
Amazing.
Thanks.
That was so